I gotta go. So today we're gonna look at amino acids and proteins. So what are proteins? Proteins are biological macromolecules. These are gonna act as enzymes, hormones, receptors, channels, transporters, antibodies, and support structures inside and outside of the cell. Proteins are compounds composed of carbon, hydrogen, oxygen, and nitrogen, which are arranged as strands of amino acids. These are going to play an essential role in the cellular maintenance, growth, and functioning of the human body. So there's going to be 20 amino acids, and these are known as the building blocks of proteins. And this is going to be the basic structure of the amino acid. So first we have the alpha amino group, which is H2N, and this is going to be attached to a tetrahedral alpha carbon, and this alpha carbon is attached to a carboxylic acid. It also has, the alpha carbon also has an R group and a hydrogen attached to it. So this R group right here is going to vary for every amino acid. The alpha carbon is attached to 4 different groups. It's attached to a hydrogen, R group, an amino group, and a carboxylic acid. So this is going to make it a chiral center. Chiral center is a molecule that has optical activity and can rotate in a plane. But not all amino acids are like this. For an example, glycine. Glycine is going to have 2 hydrogens attached to it, so therefore it's not going to be a chiral center, and it's going to make it achiral. We'll look at glycine in a little bit. I just wanted to restate how the R group, how the alpha carbon can be a chiral center for all 19 of the amino acids except for glycine. And the R group that we're just looking at, this is going to be a unique feature for every amino acid. It's going to make every amino acid different, and it's going to give its physical and chemical properties. Also, I want to make note is that all 20 amino acids are going to have this same backbone. And this is going to be a nitrogen, carbon, carbon backbone. Now we're going to look at Fisher projections of amino acids. So the Fisher projection is going to look something like this. We have the carboxylic acid attached to your carbon, attached to a hydrogen, your R group, and your amino group. And we can actually look at these across from a mirror. We can put a mirror, and we can see that these amino acids are going to be non-superimposable. So what does non-superimposable means? It means we can't put this left group onto this D group. We can't place one on top of another because it's going to give the same molecule. Therefore, these both are going to be enantiomers. So if we look at the left side, this is going to be an L-amino acid. These are constituents of proteins. So the body synthesizes most of its L-amino acids, and these get incorporated into proteins. As for the right side, this is going to be labeled as D. D means right side for the amino acid. Our body also contains D amino acids, but they're very uncommon. Our body will prefer more of L amino acids rather than D per se. So moving along, we're going to actually take a look at each amino acid separately. We're going to just go over the structure, the name, the abbreviations, and then like if it's polar, nonpolar, neutral, acidic, basic. And then we'll just give a little bit of information, extra information, just to help, the mind and neurons trigger different things for each amino acid. But before we go over that, we're going to go over like what hydrophobic means, polar amino acids, the hydroxyl groups, acidic amino acids, basic amino acids, and sulfur-containing amino acids. So we'll go over that real quick. So hydrophobic nonpolar. So these amino acids that are going to have the R, an alkyl, their R group is going to be either an alkyl or an aromatic group. So it also can be aliphatic or aromatic side chains. But these hydrophobic residues are associate with each other rather than water. So these are not going to like water, and they're going to be found on interior of folded globular proteins. So for the alkyl hydrophobic nonpolar amino acids, it's going to be glycine, alanine, valine, leucine, isoleucine, methionine, and proline. And for the R aromatic, it's going to be phenylalanine and tryptophan. So these are going to be the hydrophobic nonpolar amino acids. Now we look at the polar amino acids. So their R group is polar enough to form hydrogen bonds with water, but not polar enough to act as an acid or base. So these are going to be considered neutral. And these are hydrophilic, which means they react with water whenever possible. And we'll actually have some hydroxyl groups, so OR attached to OH. And these are going to be serine, threonine, and tyrosine. These all have hydroxyl groups that are often modified by attachment of phosphate group by the regulating enzyme kinase. The structure changes because phosphate group is very hydrophilic, and this is very important for regulating proteins. So the neutral hydrophilic polar amino acids are going to be tyrosine, serine, threonine, cysteine, glutamine, and asparagine. Now we look at the hydrophilic polar acidic, and there's only going to be two amino acids in this category, and that's going to be glutamic acid and aspartic acid. And these are the only amino acids with a carboxylic acid attached to their R group. And their pKa is going to be equal to 4. Now we're going to look at the basic amino acids. And the basic amino acids are going to be lysine, arginine, and histidine. Lysine has a pKa of 10, arginine has a pKa of 12, and histidine has a pKa of around 6. Histidine, this is going to... So now looking at the basic amino acids, there's lysine, arginine, and histidine. Lysine has a pKa of 10, arginine has a pKa of 12, and histidine has a pKa of 6. So histidine is a very unique amino acid because because the pKa is so close to physiological pH that at pH 7.4, histidine can be in a protonated or deprotonated form. And a good way to remember histidine is that his goes both ways. And the whole protonation and deprotonation, this basically means that it's readily available for proton accepting or donating. So the sulfur-containing amino acids that actually are going to contain a thiol, which is an SH group, and that's going to be cysteine, which is fairly polar. And then there's methionine, which is a thiol ether, and this is nonpolar. And two other unique amino acids, proline, and I've talked about glycine. These are basically going to create kinks in secondary structure in regards to the alpha helix. So proline this actually has a secondary alpha amino group. So right here, we could see it's a secondary. And with this group, this is what's going to actually create a kink in the alpha helix. And they're both known as alpha helix breakers. And before we start going exactly over every amino acid, we're going to name the 8 essential amino acids. And this is going to be valine, leucine, and isoleucine. These are known as branched chain amino acids. And then we also have phenylalanine, tryptophan, methionine, threonine, and lysine. So first, let's start going through the hydrophobic nonpolar amino acids. So first, we'll start off with glycine. Glycine. And this is going to be the structure. So we have H2N attached, your alpha amino group attached to your alpha carbon, and then your carboxylic acid, and then you're going to have your R group, which is just going to be an H. And the symbols and lettering for glycine are going to be GLY and G. And glycine is very important for muscle degeneration by supplying additional creatine, compound present and this is a compound present in muscle tissue. This also improves glycogen storage, freeing up glucose for energy levels, and is essential for synthesis of nucleic acids. Glycine also functions as a neurotransmitter and can help prevent epileptic seizures. So now we have alanine. And we don't have to change. Now we're going to look at valine. So valine is known as Val, V-A-L, and V. And this is going to have a CH, CH3, 2 group attached to it. And this is a nonpolar hydrophobic. Its stimulant effect has a stimulant effect, so it's needed for muscle metabolism, tissue repair, and maintenance for proper nitrogen balance in the body. It's a branched chain amino acid and used as an energy source by muscle tissue. Now we have methionine, and this is going to have a CH2, and this actually has an S, which makes it a sulfur containing amino acid, just like cysteine. But this is actually hydrophobic and nonpolar. And the abbreviation is going to be MET and M. And this is an essential amino acid that helps break down fats. This can help prevent buildup of fat in the liver and arteries that abstract blood flow to the brain, heart, and the kidneys. This can convert methionine into cysteine, a precursor of glutathione. And this is a key neutralizer of toxins. And this is not synthesized by the body. So now we're going to go ahead and look at leucine. So leucine, its R group is going to be CH2CH with CH3 2. And it's going to be abbreviated as LEU and L. And this is hydrophobic nonpolar. This is also an essential amino acid. It's branched chain. It's a branched chain amino acid, and this protects muscle, acts as fuel, protects and promotes healing. It's recommended for after surgery, and aids in increasing hormones. Next, we'll look at isoleucine. And this is going to be CH2, CH, CH3, 2. And this actually, their abbreviation is going to be I, L, E, and I. And this is needed for hemoglobin formation, stabilizes and regulates blood sugar and energy levels, and it's metabolized in muscle tissue, and this is also a branched chain amino acid. Next, we look at proline. And proline, like we talked before, is very special. It's going to be very unique. So let's erase all of this. So this is going to be the formation of proline, which has the secondary alpha amino group. We have the NH, and this is going to be abbreviated by PRO, P-R-O, and P. And proline improves skin texture by aiding in production of collagen and reducing loss of collagen through aging. Next, we look at phenylalanine. And phenylalanine is going to have a phenyl group. So, phenylalanine. And this is abbreviated by PHE and F. And drawing this, so we have your carboxylic acid attached to the amino group. And then you have the phenyl group, which is going to be attached to your alpha carbon. And this is an essential amino acid. This can cross the blood-brain barrier, which is very important, and it has a direct effect on brain chemistry. So when this is in the body, it can be converted to tyrosine, which is polar. And this is used to treat Parkinson's disease and schizophrenia. And this typically has three forms: the L, DL, and D. And the L form is the common type. The D form acts as a painkiller, and DL combination and like D form, it can be effective in controlling pain like arthritis. The last hydrophobic nonpolar amino acid that we have to talk about is going to be tryptophan. And we're just going to completely redraw this whole structure. Alright, so let's give tryptophan a look. Tryptophan. And this is abbreviated by TRP and W. So we have your carboxylic acid, okay? And this is attached to the amino group. And then here, H. And these are going to be aromatic. And this is an essential amino acid necessary for B3 production. It's used to produce serotonin, and it's a necessary neurotransmitter that sends nerve impulses from one cell to another and responsible for normal sleep. Now we're going to look at the hydrophilic polar amino acids. And these are going to be neutral. So we have serine, which is abbreviated as SER and S. And this has H2N, your alpha amino, attached to your alpha carbon, attached to your carboxylic acid. And it's going to have CH2OH. So this actually has a hydroxyl group attached to it. And this is needed for proper metabolism of fats and fatty acids, growth of muscle, and maintenance of healthy immune system. It's important in RNA and DNA and the cell membrane. Now we have glutamine. So glutamine is abbreviated as GLN and Q. And this is going to have a CH2, CH2, CO, NH2 group, R group attached. And this is the most abundant free amino acid in the muscles of the body. It readily passes through the blood-brain barrier and can act as brain fuel. In the brain, it is converted to glutamic acid. Increases the amount of GABA, which is needed to sustain proper brain function. It's maintaining proper acid and alkaline balance. When an amino acid is broken down, nitrogen is released, but the free nitrogen can form ammonia, which is toxic to the brain tissues. Liver can convert nitrogen into urea, which is excreted in urine. Nitrogen may also be attached to the glutamic acid, forming glutamine. And this creation helps clear ammonia outside of the body. You do not want ammonia in the body. Now we have 309. And this is THR and T, and that's how it's abbreviated. And this has a CH, OH, and CH3. So this is also has a hydroxyl group, and this is proper. This is essential for proper protein balance in the body. It's important for formation of collagen, elastin, and tooth enamel. It's present in heat, in the heart, CNS, or in the skeletal muscle. Now we have asparagine, and this is ASN and N. So this has CH2, CO, NH2. And this is needed to maintain balance in the central nervous system. It prevents you from being over nervous and like overly calm. It's converted into aspartic acid, and it releases energy that the brain and CNS use for the metabolism. And there's actually two others, cysteine and tyrosine, that are polar neutral and they're hydrophilic, but I'm not actually going to go over them. And I'm also not going to go over the acidic aspartic acid and glutamic acid, and the basic hydrophilic lysine, arginine, and histidine, just because I kind of went over them and said a little bit of information and don't really need to go over them at this moment. So now we're going to look at protein structure and how it's bonded. Covalent bonds are the bonds between amino acids and proteins. There's going to be a peptide bond, which is linkage of amino acids into polypeptide chains. It's like little chains like these, alpha, or amino acid. So these are going to be polypeptide chains. And the way that they're formed, they're going to be formed by an alpha amino group donating electrons to the carboxyl group, and there's going to be a loss of water. So we'll draw this up very quickly. So here, this is where the alpha amino group is donating electrons to the carboxyl group. And then we see the loss of water in this process. And this is going to be known as a nucleophilic elimination reaction. And then this is how the amide or peptide bond is going to look. And each amino acid is known as a residue. So this part right over here, this is a residue. And then this side is a residue as well. So question here is, an oligopeptide PHE is attached to glutamine, attached to glycine, attached to serine, and alanine. What number of acid and base are in this? There's going to be glutamine only, because glutamine is the only acid present. And then which residue has a free alpha amino group? And that's going to be phenylalanine, which is the first group. And then there's going to be a free carbonyl group at the end. So we'll just draw this out really quick to show you. And I'm not going to draw up any on the thermodynamics, but when we have a chart, there's 2 amino acids at the bottom, and then there's free energy going up, the reaction coordinate, and then it goes up, and then transition state comes down into a polypeptide or dipeptide. So what does thermodynamics exactly state? This states free energy must decrease to proceed spontaneously, and that such a reaction will spontaneously move toward equilibrium. At equilibrium, which is thermodynamically favored, dipeptide or amino acids? Well, since dipeptides have a higher free energy, it's going to be less favorable, and the existence of the chain is less favorable than the existence of isolated amino acids. How are peptide bonds formed and maintained inside the cell? Protein synthesis causes storage energy to force peptide bond formation, and once the bond forms, it actually its destruction is thermodynamically favorable. It remains stable because activation energy for hydrolysis reaction is so high. Hydrolysis is thermodynamically favorable but kinetically low. So what is the exact hydrolysis of a protein? There's actually acid hydrolysis and proteolysis or proteolytic cleavage. The acid hydrolysis is combined with heat and is nonspecific, and proteolysis or proteolytic cleavage, this has specific cleavage, and this protein that does the cutting is known as protease. So the enzyme that does the cutting, it has, there's a specific cleavage. It's specific to an amino acid, and it's going to be known as protease. And there's also many enzymes that only cleave the peptide bond adjacent to a specific amino acid. So if we take protease trypsin, this typically will cleave on the carboxyl side of positively charged residues arginine and lysine, as chymotrypsin cleaves adjacent to hydrophobic residues such as phenylalanine. And then based on the information above, if the following is cleaved by trypsin, what amino acid will be on the new N-terminus? So this is what we're talking about. 2N. So we have alanine, glycine, glutamine, lysine, phenylalanine, and lysine. So there's going to be two fragments And then it's going to be phenylalanine attached to phenylalanine and attached to lysine, and then the other fragment, alanine attached to glycine, attached to glutamine, and attached to lysine. Now we're going to look at the disulfide bond. So the disulfide bond is created by cysteine, which has a very reactive thiol sulfohydryl SH group, and the R group. Thiols react with each other, and if two cysteine are close together, they react, forming a disulfide bond. And the disulfide bridge plays an important role in stabilizing tertiary proteins. And this disulfide bond is actually known as cysteine, and this is spelled with a Cystine. And the reducing form is going to be when cysteine is actually just in regular form next to each other, so they don't lose the hydrogens, they don't lose the protons. A way to remember this is redox reactions. It's oil rig, and oxidation is lost and reduction is gained. And the disulfide bridge is actually oxidizing. So disulfide bridges form extracellular, or the exterior of the cell, because it is in an oxidizing environment. As the cysteine, Cysteine, that aren't bonded are in the intracellular, or the inner cell, because its antioxidants make it a reducing environment. Now we look at the primary structure. The primary structure is the amino acid sequence. This is linear ordering of amino acid residues, and it's a linear sequence. Secondary structure, this is where hydrogen bonds are between backbone groups. This is the initial folding of the polypeptide chains, and they're stabilized by hydrogen bonds between NH and CO groups. There's the alpha helix. This has the same dimensions. The alpha carboxyl oxygen of one amino acid residue is hydrogen bonded to the alpha amino proton, and proline glycine form kinks in the polypeptide chain and are never in alpha helix. There's also a beta pleated sheet, and this is stabilized by hydrogen bonding between NH and CO. It occurs between residues distant from each other. The backbone is extended, and then there's parallel and anti-parallel. So we typically find hormone receptors in ion channels found in the alpha helical transmembrane regions, integrated into the hydrophobic membranes of the cell. Alpha helix favors the hydrophobic transmembranes because all polar NH and CO groups are hydrogen bonded on the inside of the helix and don't interact with hydrophobic membrane interior. Now we look at tertiary structure. Tertiary structure is hydrophobic, hydrophilic interactions. These are located more distantly from each other in the polypeptide chain. Many different folds occur. Folding of alpha helices from secondary structures into higher order tertiary structures. These are driven by interacting of R groups and water. Hydrophobic R groups fold into the interior as hydrophilic fold out and are exposed to water. The disulfide isn't a great example because of its covalent bond, but it's still considered part of the tertiary folding. Quaternary structure, this is various bonds between separate chains. The interactions between polypeptide subunits. A subunit is a single polypeptide chain that is part of a large complex containing many subunits. Arrangement of subunits in a multi-subunit complex. So this is the quaternary structure. This has the same forces as secondary and tertiary that stabilize its structure. Non-covalent bonds, hydrogen, van der Waals interactions, and polypeptides aren't involved in quaternary structure. Now looking at the last thing is denaturation of proteins. Each protein is folded into three-dimensional structures that is required for protein to function properly. Denatured is the unfolding, improperly folded of proteins. So this refers to disruption of a protein shape without breaking peptide bonds. And what can proteins be denatured by? Urea. This disrupts hydrogen bonds. Extremes of pH, temperature, and changes in salt concentration.